Hello, everyone, and thank you to the organizers for inviting me to contribute to this important event. I'd like to extend my appreciation to the Arab Organization for Agriculture Development, the World Organization for Animal Health, and the WICA Center for their dedication to advancing brucellosis control and prevention in the Arab region. I'm very happy to present Starida's research priorities for brucellosis. First of all, let me introduce you our organization, STAR-IDAS, which is an international research consortium and is a platform for global coordination of animal disease research. STAR-IDAS stands for Global Strategic Alliances for the Coordination of Research on the Major Infectious Diseases of Animals and Zoonosis. It is a network of vendors and program owners aiming to maximize the impact of animal health research funding through coordination and cooperation. The aim of the consortium is to improve coordination of research activities to speed up the delivery of improved control methods. As you can see from the map, 35 partners from 23 countries in different regions of the world, which is highlighted in dark blue, decided to join into an international consortium, and alone they move more than 2.5 billion of USD investment on animal health research. The STAR-IDAS network connects much more countries than the ones of partners, as several additional organizations in more than 55 countries, which is in light blue, that are expressed interest in our activities and participate in our regional network meetings. Of course, new partners are always welcomed and joining is easy. In STAR-IDAS, we have at least 30 priority diseases, including brucellosis. Regional networks for Africa and the Middle East, Americas, Asia and Australasia, and Europe. They meet every 6 months by webinar in the objective to update members on activities of the global network, to discuss and agree common research priorities for their region, to explore the opportunities for sharing resources, including access to samples or strains, facilities and expertise, and lastly, to identify international funding opportunities. This slide is showing the consortium organizational structure. On the top, you can see the regional networks and the IRC Executive Committee, which is included representatives of research funders and program owners who have signed the letter of intent to align their research programs and address the gaps identified in the roadmaps by the working groups and prioritized by the scientific committee. Next, we have the scientific committee, which is including 16 independent experts covering a range of fields and are responsible for guiding the outputs of the working groups and making funding recommendations to the executive committee. In parallel, we see the CIRCA, which is the Secretariat for the consortium, and it involves many organizations, including CREAVET, BBSRC, CABI, DEFRA, and WOAH, and the Secretariat is funded by the European Commission, and its role is to support and facilitate the activities of the consortium, including the executive committee, scientific committee, and the working groups. And lastly, we have the working groups, which is including or brucellosis working group, and it involves researchers that are tasked with performing research gap analysis to feed into the research roadmaps, which will be used to advise the executive committee on where to direct their funding. As today we are in the Middle East, here are the list of the Executive Committee partners from Africa and Middle East region. And it includes Kenya Agriculture and Livestock Research Organization, l'Office National de Sécurité Sanitaire des Produits Alimentaires from Morocco, Nigerian Animal Health Research Network led by National Veterinary Research Institute from Nigeria. From Tanzania, we have the Tanzania Veterinary Laboratory Agency. From Uganda, we have the National Agricultural Research Organization, and we have international organizations like WOAH, International Livestock Research Institute, Bill and Melinda Gates Foundation, Diagnostics for Animals, Health for Animals, and Zoetis from industry, and other organizations. You may notice that within the Africa and Middle East regional network, most the partners are African countries. So I will take the opportunity of today to invite Middle East countries to join the consortium. We all know that brucellosis is a significant zoonotic disease that affects livestock and humans globally. And for this, Stardust is dedicated to addressing these challenges through structured activities. Stardust developed a roadmap for vaccine development. This roadmap guides the research and development of effective and safe vaccines to prevent brucellosis. By coordinating international efforts, Stardust aims to advance the creation of vaccines that can be widely implemented to control and eventually eradicate the disease, improving both animal and public health. The roadmap for the Brucella vaccine highlights several important areas that need to be focused on for the development of effective vaccine. These areas include the creation of vaccine that can distinguish between infected and vaccinated animals, enhancing vaccine safety to prevent interference with diagnostic tests, and the creation of vaccines that offer sterile immunity. In addition, there is a need for a better understanding of the interactions between Brucella and the host, identification of protective antigens, and overcoming technological challenges in vaccine delivery systems. Addressing these gaps is crucial in advancing the development of safe and effective Brucella vaccines. If you noticed, the roadmap was published on 2018. And to advance brucellosis research, we are about updating the vaccine roadmap and creating new roadmaps for diagnostics and control strategies. This process started with insights from the Discontrols report. Also, we created a survey that will be shared among over 300 experts in brucellosis to gather input on research needs. Data from the survey will be analyzed to identify gaps, leading to a gap analysis workshop that we are planning to organize alongside the Brucellosis International Conference in November 2025, fostering global collaboration and setting future research priorities. Here is a summary of Stardust and Discontrols' main research priorities for brucellosis. We know that effective diagnostics are crucial for early detection, accurate treatment, and controlling the spread, especially in regions where resources are limited. Here are the key areas where we need to focus to improve diagnostic tests and tools. First, availability, ensuring that diagnostic kits are accessible in resource-limited regions such as Africa, Asia, Middle East, and Latin America is essential. We should aim to validate and reduce costs for existing tests like PCR and serological assays such as ELISA and Brucellosis to make them more widely affordable. Next, standardization. Right now, diagnostics vary widely in accuracy across regions. Aligning these tests with international standards like those from WOAH will help provide consistent, reliable results and support international trade in livestock. Lastly, we need species-specific diagnostic kits for exotic and wildlife species such as camelids, water buffaloes, and yaks. Validated tests are still lacking. By creating standardized kits for these animals, we can improve detection and management in a wide range of species. Our next priority is vaccine development. Brucellosis vaccines are essential for prevention, but current options have limitations, especially regarding safety and suitability across all species. Here are the main focus areas for vaccine research and improvement. Firstly, safety. Existing vaccines like S19 and Rev1 are effective but can pose risks, particularly in pregnant animals. We need vaccines that are both safe and effective across all stages of animal life, reducing adverse effects while ensuring strong immunity. Second, we need to focus on DIVA, or differentiating infected from vaccinated animals vaccine. These specialized vaccines would allow us to distinguish vaccinated animals from naturally infected ones, which is especially valuable in regions using combined testing and vaccination programs. Lastly, species expansion. Currently, we lack brucellosis vaccines for certain species, especially some wildlife. Developing vaccines specifically for those animals would allow us to protect a broader range of species, especially in regions where these animals play a role in the disease cycle. So, in short, enhancing vaccine safety, expanding to new species, and developing DIVA capabilities are crucial steps to improving brucellosis prevention worldwide. Our third priority is strengthening surveillance and control systems for brucellosis. Effective control relies on comprehensive data, secure traceability, and coordinated action across sectors. Here is how we are addressing these needs. First, data collection. Standardizing surveillance methods and collecting reliable data on brucellosis prevalence and economic impact are essential. This data will guide policymaking and ensure resources are directed where they are needed most. Next, biosecurity and traceability. By establishing strong biosecurity protocols and animal traceability, we can control the spread of brucellosis more effectively. This is especially important in regions where animal movement is common and disease spread is a risk. Finally, the One Health approach, integrating surveillance across animal, human, and environmental health sectors, allow us to monitor and respond to brucellosis more holistically. And this collaborative approach is crucial for long-term sustainable control. So together, these measures will enable us to better monitor, manage, and ultimately control brucellosis. These research priorities are directly informed by the main conclusions of recent studies on brucellosis. And here are three key findings that highlight the ongoing challenges and impacts of this disease. The economic burden. Brucellosis significantly impacts livestock productivity and trade, creating economic challenges, especially for agriculture-dependent regions. Public health risk. As a zoonotic disease, brucellosis poses risks to human health, particularly in areas lacking resources for effective control and prevention. And lastly, resource gaps. which is a major obstacle in brucellosis management, is the shortage of trained personnel, lab facilities, and funding, which limits the effectiveness of disease control efforts. After identifying our priorities in diagnostics, vaccines, and surveillance, specific actions need to be implemented. And here are core recommendations. First, capacity building. We recommend engaging in training programs such as those offered by WOAH to improve diagnostic and surveillance capabilities, especially in regions where resources are limited. Second, developing strong policies and legislation is crucial. Implement the regulations on farm registration, animal traceability, and biosecurity will help prevent and control brucellosis more effectively. This structured approach will also support better disease management in international trade. Third, we emphasize One Health approach, encouraging coordination across animal health, human health, and environmental sectors will ensure a comprehensive response to brucellosis and other zoonotic diseases. This multi-sector collaboration is essential for sustainable disease control. So together, these recommendations will strengthen our capacity to monitor, manage, and reduce the impact of brucellosis globally, aligning with our broader priorities. Thank you for your attention, and for any questions or inquiries, please feel free to reach out. If you are interested in joining the Staritas Brucellosis Working Group or other priority disease working groups, please contact me or my colleague Yohanna Shahriar from CRIAPER.